Hello, and welcome to Part 2 of Chapter 11 on Mendel and the gene idea. Now, we've been talking mostly about these Mendelian traits. So Mendel had his pea plants, and he looked at characteristics which had binary traits, purple versus white flowers, yellow versus green peas. And this was a real advantage for his understanding of how heredity works. But we know that not all characteristics are inherited following this exact same pattern. So we can really add to what Mendel did here. And so here we have Mendel's assumptions, single genes, one characteristic, one gene, only two alleles. There's also a clear dominant recessive relationship that we saw. But really, most genes don't meet these criteria. So again, Mendel may be a bit lucky or careful in his choices of his pea plants and the characteristics that he looked at, because so many characteristics don't. So let's look at a list of these examples. Codominance. We've already seen this. This came up when I talked about blood type. So codominant situation here would be the AB blood type. So this individual has a IA-IB allele, so two alleles, the A and B, and both of those traits are expressed. So this is codominance. One has not masked the other. So if A is present, it doesn't hide B, it doesn't mask them. They're both present. Another example, incomplete dominance. Again, we've seen both of these before. This was the case where this looks a little bit like blending. The heterozygote here has an intermediate flower color. It's not red, it's not white, but it's in between. So that would be a case of incomplete dominance. Another extension or exception to some of these Mendelian patterns would be genetic polymorphism. And we can use blood type yet again to illustrate this point. Now, for the ABO blood type, there are three different alleles, IA, IB, and little I. Any individual only gets two of them, but in the population, there are those three alleles. You know, some genes might have many alleles. And so that is called polymorphism. Poly means many, morph means shape, but really the idea is poly, we've got many alleles. So let's look at a couple of more examples of those. So coat color in animals is a great way to illustrate this. And here in rabbits, so rabbit coat color, there are 4 different alleles. And we also have an interesting situation where some alleles are dominant, some are incompletely dominant, some are recessive, and it depends on the combination that you get. So this C plus allele, this is the agouti, so here's an agouti colored rabbit, that is dominant over all others. So if any rabbit has this one allele and any other, maybe another C plus, maybe any of these others, they're going to have this appearance. Let's see, the chinchilla CH allele. Of course, it's recessive to C plus, right? Because C plus is dominant to everything. But it's only incompletely dominant over these other two here. So you might end up with intermediate characteristics or intermediate coat colors if you have those combinations. If it's CCH and CCH, a homozygote, it'll be the true chinchilla color. The Himalayan rabbit we see is recessive to everything above, but certain dominant over this last allele, the albino allele. The only way to get an albino rabbit would be to have 2 of the C alleles here. All right, so here's another example, mouse coat color. So this is an example of a dominance hierarchy. So any of these alleles down the list or up the list, let's say, is dominant over any of the other one below. So I'll give you a genotype here in the question. What would the coat color of this mouse be? Okay, yeah, you're just picking the dominant allele and going with whatever color that confers. This other allele, if it's lower down on the list, just doesn't matter, doesn't contribute to the coat color. It might be important for next generation, but in that individual, it's the dominant allele that will determine the color. How about a mouse that is AY, AY? So 2 of these yellow alleles. What color is that? Now, it might seem obvious, well, that's going to be yellow, right? Interestingly enough, no, it is a dead mouse. You cannot get a true breeding line of this YY. I've just cut out the A here, just using Y for yellow. So Quinault was one of the early pioneers in studying mouse genetics and this kind of heredity, and he was trying to get a true breeding line, right? Because you need a true breeding line of something to do your experiments. But he could never get that. And this illustrates another point that is important, and this is called pleiotropy, the idea that an allele or a gene can have more than one effect. So this allele for coat color also has some other function in this mouse. So if you're missing, or if you have two of these Y alleles, that is not going to be a viable offspring. A mouse can't survive. It needs at least one of these other alleles for coat color. So again, that means these are important for not just for coat color, but some other function, and that's pleiotropy. Pleo sort of means like plastic. And so basically the idea that an allele has more than one impact. The next extension to Mendel is called epistasis. Now, epi means upon. And so this idea here is that you have one gene acting upon the expression of another gene. So this is different from pleiotropy, where we're talking about one gene having multiple effects. Here we have two, maybe at least two different genes interacting, one acting upon epi, the other. And a great example, because here we got these cute little dogs here, is coat color in Labrador retrievers. So there are two genes associated with coat color. One is this E gene. So if an individual has capital E at all, then that will produce a dark colored dog. The only way to get a yellow lab is to have little e, little e. Now, the next part here where we have the interaction between two genes is this B gene. If an individual is dark coat color, one of these genotypes, then that B gene will determine how dark. So if you have at least one big B allele, that's going to produce a black lab. If you have both little b's, that will produce this brown or chocolate lab. And we could plug that into a Punnett square if you wanted and see what the outcomes look like. So here we have all the different gamete genotypes and then the possible outcomes that would be produced from these. Another example of epistasis is feather color in chickens. So there's two genes involved. One is this C gene. This produces a pigment. So the way to get a colored, dark colored chicken is you'd have to have the C, capital C allele. So that's step one. So here we see a dark colored chicken, good, capital C allele. But look at this guy. It does also have a capital C allele, but why is it a white chicken? It's because this other gene is interacting, the I gene. This is an inhibitor, for I for inhibitor. If a chicken has the capital I allele, that will produce a protein that interacts, stops the expression of pigment, production of pigment. And so you can see that here. The only way to get a pigmented, dark pigmented chicken is if you have two little I alleles. So that bird is not producing the inhibitor, and therefore that pigment can be expressed. Now, it doesn't matter down here. This white colored chicken here, it has two little I alleles, yes, but look, it doesn't have the capital C allele, so it doesn't produce pigment. So that is an example of epistasis. Now you try one. Our next extension to Mendel is polygenic inheritance. And this relates to epistasis, which we just talked about. The idea that multiple genes control a phenotype of a trait. Now, this is usually applied to something like, say, human height, for example, where we're looking not at discrete variation, like purple versus white flower, but we're talking about a continuous variation. So people are not just 4 feet or 5 feet tall. There's a continuum. these are referred to as quantitative traits. And so there's multiple alleles involved. That increases the number of potential outcomes. And as we'll see in the next phase here, or next extension to Mendel, environment also plays a role. Human skin color can be considered a quantitative trait. Now, if we just look at human skin color as a product of 3 genes with these different alleles, we can see a gradation of different skin colors in this example, and you come up with all the probabilities that produce all these different categories. But if you think about it, skin color is not a binary trait, certainly, and there are more than the 7 phenotypes that are listed here. So what are some other factors, or what's another factor that might influence human skin color? Right. A lot of different things can influence human skin color, and the environment plays an important role. So that's our next extension, this idea that gene expression may be influenced by the environment. Now, these are examples where we have more of a direct influence. So this little kitty cat here, and we see this in Himalayan rabbits and Siamese cats, this dark coloration out in the face and in the ears and the tips of the tail and in the extremities. And what's happening here is this gene that produces this pigment is only active below 33 degrees. So, why is it out here in the tail and the ears? Is because these are parts of the body that are slightly lower temperature, right? So that allows for the expression of this gene and the production of this pigment. So that's one gene being influenced by the environment. Another example of environment interacting with gene expression is flower color in these hydrangeas. So in more acidic conditions, the flower color will be this beautiful deep blue color versus, say, pH 7 soil. You get these much more pink blooms. So you can actually influence the flower colors that your hydrangeas will produce depending on the acidity of your soil. Now getting into maybe more complex things where we're not just dealing with single genes, the interaction of the environment and the genome in general. So what I'm showing you here is a picture of 2 different genetic lines of mice. We're calling them A and B. Now, if A and B are fed a typical mouse diet, so mice from these different lines, they end up looking pretty similar. They weigh about the same, they look very similar. But when we switch these different genetic lines of mice to a high-fat diet, for example, this genetic line A tends to take on that fat, whereas this different genetic line, it's eating the same diet, the high-fat diet, it doesn't accumulate as much fat on its body. So this shows it's not just what the mouse is eating that's important. There are underlying genetic factors that are associated with that outcome. A really cool way that we can explore some of these genetic versus environmental differences is looking at twin studies. Now, you have different kinds of twins possible, right? So fraternal twins, those are twins that are born at the same time, but they originated from 2 different fertilization events, whereas identical twins are genetically identical, originated from the same fertilization event. So what would we expect when we look at some, in this case, disorders or diseases in twins, identical versus fraternal twins? If something is purely genetic, we'd expect a very high concordance in identical twins, but less so in fraternal twins. And so let's take an example here. Say high blood pressure. Look, 25% of identical twins both have high blood pressure. That's not really a huge number. This suggests that, probably environment has a lot to do with high blood pressure. There may be some genetic component to this, but it maybe isn't huge. Let's look at fraternal twins. That high blood pressure actually much lower. And so what this suggests is, okay, environment's important and genetics play a role because identical twins did have a higher prevalence of high blood pressure than fraternal twins. So if you're genetically identical, that was higher. That means there is a genetic side to this. let's take another example. This last one, acute infection leading to death. Okay, low number straight out of the gate, and both of them are pretty similar, 8 and 9%. This suggests that environment plays a big role in this, and that makes sense, right? An acute infection is some random event that could happen in your lifetime, whether or not you're a twin or not. Measles is similar, right? Measles is high. Look at that concordance, very high. Well, is that genetic? Well, it looks maybe like it until we look at the fraternal twins, also very high. The difference between these two is not significant. And this tells us environment plays a role. Why would these be high? Because children tend to get measles. And so whether you're fraternal or identical twin, you're usually living together and therefore more likely to get measles at the same time. So we have to take a step back and look at this really broadly. We've talked about phenotype and genotype and now the environment. And we could think about this very simply where, okay, you've got a certain genotype and given environmental condition A, phenotype A results. Or environmental condition B, you get a different phenotype. And yeah, that could be the case. And we saw examples of that, say like the flower color in hydrangeas. But as we start to look at more complex traits and an environment that is changing and different for, especially we're talking about humans, over the lifespan of a human, that these all interact. And that is what results in the phenotype. All right, so let's do a little trait tracking. Now, in humans, you can't really do the sort of pea plant type studies like Mendel did. So humans aren't really a good subject for genetic research. I mean, we're very interested in human genetics, but you can't do experimental crosses. You want statistically good numbers. You would like a lot of offspring. Usually you're not going to get that from humans. Generation time is long, so not great in that respect either. And so one of the ways that we track traits through human families is through something called a pedigree analysis. This is used in other animals and organisms as well. And when we look at pedigrees, we can often see where we can track a dominant or recessive trait through a population. And so let's look at some of those examples. Let me orient you first to what a pedigree looks like. And this is a nice example here. So we're looking at individuals represented by these shapes. So a male is typically represented by a square, a female by a circle. And so here where we have a male and female, there's a line connecting them laterally here. This represents a mating in between two individuals. And then we have a line extending down. This would be extending down to their offspring. So here we have annotated generation 1. Here's generation 2. And then the offspring here are, son, daughter, daughter, son, son, daughter, daughter. And we can see another thing appear here, these red squares and red circles. This would be an affected individual. For whatever trait we happen to be interested in or talking about, that's just a way to highlight, hey, here's where this phenotype appeared or trait appeared. Notice here, you see this red square, it's not connected to the line above. So this red square, this is not an offspring of parents in generation 1 here. This is a new individual that's appeared on our pedigree and a horizontal line connecting to this female. This male, it mated with this female, and here are those offspring. Here's an example of identical twins. That's how we annotate that. The line coming down here. Non-identical twins have a slightly straight line and then it branches. And here sometimes we see this double line, meaning mating between relatives. So that orients you to what a pedigree looks like. Let's take an example. So brachydactyly, you see the condition here in this image. And so we can trace this trait through this family. And what we see is this looks like a dominant trait. Some of the things we might take note of is it does appear in every lineage, every generation, I should say. So it appears commonly. That doesn't mean a recessive trait can't appear commonly, but Usually, if we see something appearing like this a lot, we're thinking this is a dominant trait. What else do we see? Here's a key point. Where we have an affected parent, about half of the offspring are affected. Now, again, you'd have to have a lot of children to get a good statistical number here, but think back to that Mendelian inheritance. This individual would have at least one of the affected alleles, and so the likelihood of them passing it to their offspring is about 50%. Unless they have two, then the probability of passing it to the next generation is 100%, and you'd see all individuals affected. But so we can assume this person was heterozygous, and half their offspring ended up with that dominant allele. All right, how about heredity of a recessive allele? So a couple of things that we might pay attention to. Well, the trait doesn't appear as often as we saw with the dominant trait. We see it skipping generations. So if this was a dominant trait that appeared in subsequent generations, it wouldn't skip a generation. And we would only see that where the trait is quote unquote hidden in a recessive allele. It's kind of like the white flower color, say, for example. We see it disappear in that hybrid generation, but then reappear in the next generation. And so for a recessive trait to be expressed, that individual would need to have both of the recessive alleles. So that's less likely, possibly less likely, and therefore we don't see it as much in this pedigree. All right, so you can ask yourself, looking at this figure, is this a dominant or a recessive trait, widow's peak? Now, the genotypes are given here too, so that should give it away. And you'll see big W, little w, this is an affected individual. This person has the widow's peak. So even a heterozygote has the widow's peak. This means it's a dominant trait. How about the ability for an individual to taste this PTC chemical? Again, we have the genotypes written here to help you along. Notice the only time we get someone who is affected is when we have the recessive homozygous genotype. All right, you try one now. So here we have a pedigree for a family, some of whose members exhibit a trait W. Affected individuals are indicated by a dark circle or square. What's the likelihood that the offspring of 4-3 and 4-4 will have that trait? So here we have the generations. 4-3, that's this individual, and here's 4-4. So there's a mating here. What are the odds that their offspring will have this trait? All right, a few things to pick up on. One is this is a dominant pedigree. Notice this individual is heterozygous and they have the affected trait. So that means our individual down here, generation 4, individual 3, this male, it has that trait. And how do we know if they're heterozygous or homozygous? We can go back to his parents. Father was affected, mother was not. So the mother must have been little w, little w. That means that the only allele that this person could have got from their mother was the little w, meaning they would be heterozygous. Their mate here, individual 4-4, would be homozygous recessive, right? The little w. And so you can plug that into a Punnett square and come up with 50% of the time, or there's 50% chance that their offspring would be affected. It's a dominant trait. Let's look at recessively inherited disorders in a little more detail. There are many examples of disorders that are recessive. They might range in severity from mild to life-threatening. And we can introduce another term here, this idea of carriers. Sometimes we would note carriers on a pedigree with this two-colored circle or square. So it's really meant to indicate this person has one of the recessive alleles and one of the dominant alleles, that they're a carrier. So let's look at albinism as an example of the inheritance of a recessive trait. Here we have the example of two parents that are carriers, so they're not affected. And here are the possible, gametes produced by each parent, and therefore the possible outcomes of their offspring in terms of the genotype. There's a 1 in 4 chance that you would get this recessive homozygous individual offspring. And so that's a 25% chance of producing a child who has albinism from these carrier parents. So the other thing to understand with recessive traits is if a recessive allele is rare, and it isn't always, just because something's recessive doesn't mean it's rare, but if it is, and that is often the case with something that confers a disease, because there's so much selection against it, the chance of two carriers meeting and mating is low. It doesn't mean it's not possible, but it's low. Where it becomes a concern, could become a concern, is where you have close matings between relatives. So these consanguineous, that basically means the same blood. I mean, obviously we're all related as human beings at some extent, but we're talking about close relationships, matings between cousins, for example. And so you can see that here. We've got grandparents in this pedigree. They produce children that are carriers. If those folks go off and here are their mates, and if that's the end of it, that trait might just get diluted or die out in that lineage. But no, in these cousins, which are both carriers, there's a mating here, and that's where we bring those rare alleles back together, which again, otherwise just by chance wouldn't have come back together. Again, this doesn't mean that every time you see a recessive characteristic appear that there's been a close mating. It's just the probability is much higher when you have mating between close relatives. Let's look at another example here, sickle cell disease. This is a really interesting one. We'll actually come back to this later in the course, very likely. So sickle cell disease affects about 1 out of 400 African Americans. So we're talking about a population here in the United States. The disease is caused by a mutation that is found, or a single substitution, let's say that. This isn't a mutation that's just happening. It's happened a long time ago. And so there's this variant, this allele that has this mutation. And this results in a modification to hemoglobin. So this is in the hemoglobin gene. That's that's a protein found in your red blood cells, important for carrying oxygen. And so this change in this protein, as we'll see, can have a big effect. So what are the symptoms of sickle cell disease? Physical weakness, pain, organ damage, stroke, and paralysis. Somebody who is homozygous for this sickle allele, without treatment, that's usually fatal at a very young age. So what's happening here? Here we have hemoglobin. It's a tetramer, so it's made out of 4 polypeptides. So it has that quaternary structure. Here's normal hemoglobin, kind of have this globular shape, hence the name hemoglobin. But here's the abnormal hemoglobin. That beta hemoglobin that's produced from the beta hemoglobin gene is slightly different based on that one mutation. And what can happen under the right conditions is it will form stacks. They'll connect on each other, form these long rods, and that will cause these red blood cells to sickle. So instead of having these nice biconcave discs, you end up with these sickle cells. Those can get caught up in capillary beds and cause all kinds of problems like we described before. So when do we see the sickle cell trait? So we can take a couple of alleles as an example. We're going to call this big H. We're saying hemoglobin. That's the normal hemoglobin. So those are individuals that are not affected and not carriers. Individuals that are big H, little h, they're not affected by the sickle cell disease, or mildly so, but they're carriers. Individuals that have little h are severely affected in this situation. All right, so we can look at this pedigree. We don't know what this generation 1 was, although these folks had no apparent disease. Here's generation 2, so here's an affected male. Let's look at this pairing here. We have a carrier parent and an unaffected parent. Will these children have sickle cell disease? Now, I'm not asking whether they're carriers or not. Will they have sickle cell disease? Okay, that might be an easy one, hopefully. They will not. They might be carriers, but they won't have sickle cell disease because they would need both of the affected alleles, and they can only get one from this male parent. So why do we see the persistence of this pretty serious condition in the population? Why doesn't this just die out? Well, it is connected to malaria. So you might have heard of malaria. This is one of the biggest parasitic diseases on the planet. And the parasite infects red blood cells. So it's a protozoan parasite transmitted by mosquitoes. And if someone has one of the sickle cell alleles, this is an advantage. And this is called the heterozygote advantage. Individuals that have this have some protection from malaria. And so what happens when, here's the scenario where somebody is homozygous, has sickle cell disease, under low oxygen condition, those hemoglobin molecules form those chains, those sickled blood cells clog up the spleen and capillary beds, and that's where you get disease. With the heterozygote, you get that too. But what might be happening, what we think is happening, is it's an infected red blood cell will form this sickle shape. Then it will get cleared out of the bloodstream. So it's like the body is selectively taking out infected blood cells. So this provides some protection from a very serious disease, malaria. So although there are problems for a homozygous person with the sickle cell trait, there is also strong selection for maintaining it in the population because it prevents or helps reduce the severity of malaria. All right, let's look at one more example of a dominantly inherited disorder. So we've seen how dominant traits are passed in a population. A lethal dominant trait is not likely to be passed if it's lethal at a very young age, because that individual isn't going to reach reproductive age, and therefore they're not going to pass it to the next generation. But if something is a lethal dominant allele, so an example is in the case of Huntington's disease, go look it up if you're interested, that tends to appear as a disease after reproductive age. So it could have been passed to the next generation by that point. A non-lethal dominant trait is achondroplasia. So this is a form of dwarfism. It's caused by a rare dominant allele, and we can see how that can be passed from one generation to the next. If one parent has that allele, there's a 50-50 chance that the children will be affected. All right. So in all of these scenarios, and hopefully you're getting this idea, whether we're talking about just phenotype or we're talking about a genetic disorder or disease, is that these things are multifactorial. So take heart disease or cancer or mental illness. There's genetic and environmental factors, lifestyle choices. All of these things connect and feed into ultimately what the outcome is. And so these are very much multifactorial. All right. With that, that ends this chapter. We'll see you next time.